Hi, my name is Noah Goldfarb. I'm an assistant professor in internal medicine and dermatology at the University of Minnesota and staff physician at the Minneapolis VA healthcare system. I'm also the lead author on our recently published study on the psychometric properties of the revised Hidradenitis Suppurativa Area and Severity Index, or HASI-R. Hidradenitis Suppurativa, or HS, is a devastating condition with painful nodules, abscesses, and draining tunnels, typically involving the armpits, chest, lower abdomen, groin, and buttocks. HS is not a rare condition. It afflicts 1 out of every 100 to 1,000 people. Unfortunately, we only have one Federal Drug Administration, or FDA-approved medication for this distressing condition. In evaluating new potential treatments, it is imperative that we have valid, reliable, and feasible tools to measure disease activity that are sensitive to change with treatment. Current validated tools available for measuring disease activity rely on lesion counts, which have recently come under scrutiny, as lesion counts are time-consuming and have been found to be unreliable. In addition, the core outcome set for HS trials calls for an assessment of body surface area, which is missing from our current validated tools available. To avoid issues related to lesion counts and incorporate an assessment of body surface area, two separate groups recently published similar tools using signs of inflammation and body surface area. One was the HS Activity and Severity Index, also known as the HASI, and the other was the Severity and Area Score for HS, or SASH. These tools were created simultaneously with the same goal, and therefore we merged the two tools to create a novel tool that addressed issues with the prior instruments. We called this tool the revised HASI, or HASI-R. This tool measures 4 domains to assess the severity of HS activity at 9 anatomic sites, including the inflammatory color change, inflammatory induration, open skin surface, and extent of tunnels. And each of these variables is scored on a Likert scale from 0 to 3, based on the average intensity for each site. The extent of HS is then assessed using BSA for each body site. Raters can estimate BSA using the entire hand area, or palm plus fingers, and then also can provide a BSA estimate to 1/10 of a percentage point using the assumption that a forefinger is approximately 0.1% BSA. The BSA score for each HS-specific site was calculated as the proportion of the site involved by HS converted to a 0 to 6 ordinal scale based on the psoriasis lattice system. To calculate the HASI-R, the site-specific score is calculated by multiplying the BSA ordinal score by the sum of the 4 intensity scores. This study was done as a step to validate HASI-R tool, to evaluate its inter-rater and intra-rater reliability, as well as convergent, divergent, and known group validity. In addition, we also received feedback from our expert raters regarding the use of this tool. In order to do this, 20 physician raters from 4 countries assessed 15 HS participants with a broad range of HS severity and affected body sites on a single day. Physician raters assessed participants using the HASI-R, Hurley staging, and lesion counts, which were later used to calculate the International Hidradenitis Suppurativa and Severity Score System, or IHS-4, the abscess nodule count, and the original modified Sartorius scores in order to evaluate the convergent validity. Every rater was then randomized to score a patient a second time at the end of the session to evaluate intra-rater reliability. HS participants also completed the Dermatology Life Quality Index, or DLQI, to calculate the divergent validity. Finally, at the end of the session, we received feedback from our expert raters. We found that the HASI-R had moderate inter-rater reliability, which was the highest value amongst the outcome measures evaluated in the study, aside from the Hurley classification at the axilla. All other outcome measures, including the IHS-4, abscess nodule count, and original and modified Sartorius scores, had poor inter-rater reliability. The HASI-R also had the highest intra-rater reliability of all tools. HASI-R demonstrated good convergent validity compared with the other clinician-reported measures and showed a weak, non-significant correlation with the reverse-scored DLQI, which is consistent with the other HS clinician-reported outcomes in the literature. HASI-R also demonstrated known group validity based on the significant difference in mean HASI-R scores amongst the established IHS-4 severity groups. Finally, our expert raters ranked the HASI-R as the highest for ease of interpreting components, ability to capture HS extent and lesion severity, as well as the ability to capture meaningful change. The HASI-R was the preferred tool for clinical trials by all raters. The results of this study demonstrate that the challenges of clinically evaluating HS by counting lesions, which has previously been highlighted in other studies. HASI-R's relatively higher inter-rater reliability, intra-rater reliability, construct validity, and rater preference suggest the HASI-R is a valid, reliable tool to measure HS severity and supports the continued evaluation of the HASI-R as a clinician-reported outcome measure. In conclusion, the use of outcome measures in clinical trials with lower reliability, higher limits of agreement, and higher minimal detectable change increases the risk that a treatment effect will be obscured or a study will get a false negative result. This can result in discarding of promising new therapies. As there is only one FDA-approved medication for HS with approximately only a 50% response rate, effective treatment options are urgently needed. In order to make sure effective therapies are appropriately evaluated, high-quality outcome measures for HS are necessary. Thank you for your time and interest in our study.